Good afternoon. I will introduce today a method for the extraction of an object in a clutter using a very known standard approach from a concept from graph theory. Now, the main character of this talk is a tiny worm, 1 millimeter long, called C. elegans, and the S here is not for plural. It is a joint work with members at the imaging platform of the Broad Institute of MIT and Harvard, and some computational and system biology labs in Harvard Medical School. So this is the worm, and it has 302 neurons, and this is why it was proposed, dated back in the 60s, as a great, excellent model for the development or the research of the development of the nervous system. Stanley Brenner, who actually proposed that, won the Nobel Prize for it. So why C. elegans? It turns out that this animal is sufficiently complex to study general principle of multi-cell systems, and at the same time sufficiently simple to be handled in high-throughput screening, that is microscopy imaging. I was always wondering how people invent new drugs. It turns out that having C. elegans, things get much simpler, at least in terms of ethics, no FDA or Helsinki. So you have millions of C. elegans, and then you infect them by a bacteria, and then you test all the known drugs and medicine you know, and just screen them after 3, 4, 6, 72 hours, and count how many dead, how many alive. Once you reduced your search space, searching for the most promising drugs, then you can move on to the next step and try it on mice. Now it turns out that it's not only the nervous system or immune system that resemble human being, despite millions of years of evolution gap. There are also other systems, such as the metabolic pathway, that can be searched and researched using C. elegans. So biologists used to knock out certain genes, investigate the relation between genotypes and phenotypes. For example, if they want to investigate fat metabolism, they can knock out genes and look for some morphological properties between and differences between controls and the mutant. So the entire process is automatic. They use robots. They can handle the preparation and the sorting and the imaging. This can be done really fast, 4 to 6 plates per hour, 384 wells. Each well contains 20 C. elegans. Where is the main bottleneck? The main bottleneck is image processing. Now, when these worms are far apart, you can easily segment them. However, they tend to touch each other, or more than that, they tend to entangle. And this turns a problem into much more a difficult one. So, here are some examples, and if you look, if I do zoom in and you look at a worm cluster, you can see it's not an easy problem. So there are some regions that you cannot tell them apart, even as a human being. What do we do to handle it? So we proposed a two-step algorithm that contained training and a test. In the training, we use probabilistic shape model. We use the medial axis transform in order to extract the worm skeleton. Having a worm skeleton, we can use PCA and extract what we called Eighen worms. Having Eighen worms, we can use them in order to define a worm likelihood model. So we have a new example, a new vector, and now we have to say how likely this example is a worm. This is not a new idea. In 1991, it was proposed by Leventon, Grimson, and Fujiras, and they use it for brain structure. They didn't use skeleton. They used sign distance function or active contour in order to do that, but we were inspired by this work. Now, the innovative part is this test one. Once we have a worm likelihood model, we would like to represent a worm cluster in a way such that we can extract individual worms. So in order to do that, we do medial axis transform, extract the worm cluster skeleton. Once we have the worm cluster skeleton, we represent it by a sparse direct graph. Now, what we need to do is to find paths in the graph that are more likely to represent worms. I will briefly go over the training. This is quite simple. We have a worm, isolated worm. We do segmentation, extract the skeleton, space the skeleton, define it as a vector. Now it's very easy to reconstruct back from the vector back to the worm. And we use it to define the model. In practice, we have a huge training set, pick all the isolated worms, put them in a matrix, and then generate what we call a covariance matrix denoted here by C. We use it to define what we call the eigenworms, UK, corresponding eigenvalues. Now, having a new example denoted here by X, we can project this new example over the space defined by the eigenworms, get the weights, and now we derive what we call the worm likelihood model, which is proportional to the exponent of the expression that depends on the weight and the eigenvalues. Okay, so we have a worm likelihood model. How this can be used in order to extract this worm cluster. And here you can see a cluster of 3 worms. Even without doing extraction, we can somehow guess or estimate that we talk about or search for 3 worms here. We simply divide the entire cluster region by the size of the mean worm. In order to do that, we take the worm cluster, and this is a very simple example of 3 worms, and generate the skeleton using medial axis transform. You can see here that there are 7 worm segments denoted by V1 to V7. There are 3 intersection points. And then if you look the edges of the graph on the right, defined by the neighborhood between 2 segments. Okay, so we moved from the skeleton to a dual representation or to the graph. Now, having this sparse graph, our problem turned to be search for path in the graph. Why did we move to the dual space? It turns out that it's much easier to handle self-intersection like this, so this turns to be a really easy graph. Back to our original graph. Searching for the most likely worms, we would like at the same time to minimize the leftover or to maximize the coverage. Now, this problem is very similar to a very known problem in graph theory of path coverage. So in path coverage, you look for the minimal number of paths in the graph that fully cover the graph. It's not this is an NP-complete problem. This problem is a little bit different. We look for k paths, not for the minimal number of paths, but for k paths, and k is the estimated number of worms, expected worms. And at the same time, we do not restrict our problem for full coverage, and we relax it and we look for the maximal coverage or the minimal leftover. Okay, so we look for simple paths in the graph. But note that not every simple path in the graph forms something that we can say it is a worm skeleton. So look, for example, on vertices 5, 6, and 7. And look how they look when we extract the corresponding skeleton. There is a branch here. That is, if in a path there are more than 2 vertices belongs to the same clique, and you can see that 5, 6, and 7 form a clique here in the graph, then this is not a valid path. This had some complexity to the problem. However, it reduced our search space, and we used that.
How do you detect the direction of the edges?
Okay, this is a good question. It's a sparse and directed graph. We give random number to the vertices, and then we just decide that we go from low number to high number. This is the direction, because it doesn't matter if you go from 1 to 2 or from 2 to 1. Okay. So we can have many types of paths. We can have a 0-length path. We can have a 1-length path. We can have a 2-length path. We can have a 3-length path. But we cannot have a 4-length path. So the maximum path length is restricted by actually the size of the click and the number of clicks. Here in this particular example, we have 7-length 0 paths, 9-length 1 path, 6-length 2 paths, 4-length 3 paths, altogether 26 paths. And we need to select the most probable 3 paths. How do we do that? Here are the example. You can see here that there is a full coverage. In this example, our vertex tree repeats. So there is some overlap, and we allow that in our problem. We can define a cost function. Okay, so the cost function is actually summing over k of the log of the minus of the log of the likelihood of being a worm, and we restrict the size of the leftover, all the vertices that have not been participated or being part of the path selected. Okay, and this is in general an n over k problem. It's a combinatorial problem, and we want to do something slightly more easier. So in order to do that, first let's define the leftover in a different manner. Okay, so what is the number of the leftover vertices? It's the total number of vertices, exists in a graph, minus all the vertices that already participated in selected path, minus the overlap. Okay, so we can put this all together, and now we can minimize this cost function. Note that we have a sum of, this can be removed because this is a constant, this is the graph cardinality, we remove it, and now for each term here, each of the 2 terms, we have sum over k. So we can write slightly differently the cost function, and it's a sum over k of this expression, which is the likelihood, the overlap, minus the length of the path. In order to make the algorithm slightly easier, we use a greedy approach. Okay, so we decompose the problem into k sub-problems, and now we solve them one at a time. So, given P1 to Pk minus 1 path already selected, the cost of additional paths Pk is minus log of its likelihood plus the overlap minus its length. The entire algorithm goes as follows. We estimate k, the number of potential paths, then use BFS to detect n paths in G, the graph. Then we define a path candidate set, P1 to Pn. And the selected path set is currently an empty set. We haven't selected any path yet. For the first path, what do? We calculate for all the n paths the likelihood minus the length. And then we pick the one with the lowest cost. So this is the first selected path. Now for all the remaining paths, we calculate the additional cost of the overlap. And then we pick again the second best path. And we go on and we go on until, for example, we already selected k minus 1 path. Now, k is just an estimate. So we want to know if we need to stop here and say, okay, there are k minus 1 worms here in the cluster, or we should go on and add more paths. So we compare 2 costs. The cost of staying here, and then we have this leftover that we denoted by v tilde k minus 1, or adding another path, path number k, and then on the cost of having minus log of its likelihood. We compare these 2 costs. We know if to continue or not. This is a stopping criteria. So there are some issues relating to computation and complexity. So the number of paths in a graph are not in the order of factorial number of the cardinality of the graph, but it's much lower, and we proved in a paper that it's in the order of L squared D, where L is the number of click and D is the maximum click size. And the selection of k paths out of n, it's not anymore n over k problem, which is a combinatorial problem, but it's in the order of n multiplied by k. Some results. So you can see here an example of a worm cluster, and you can see on the right the segmentation, and below you can see the skeleton with the intersection points, and there are many paths here in this simple example. These are some more results, but in fact, these were only preliminary results. Nowadays, the algorithm was implemented in Cell Profiler. The Cell Profiler is a toolbox. It was developed by the imaging platform, my collaboration. So cell detection, cell extraction, and now C. elegans detection is part of this module. And it's used by numerous labs around the world of system and computational biology. So we started with worms, and we ended up with graph search. And I've been asked if this work can be applicable to real-world objects by the CVPR reviewer who decided to reject this paper because it was not general enough. Obviously, that was a rhetorical question. So I believe that it might be with the right likelihood model or shape model. Maybe using graphs can be inspiring to solve some other problems. And that's it.